Good evening, everyone. I am recording the pre-lab video. This is going to be about half an hour, and it's pretty late, but I want to make sure that we get this out to you by Tuesday morning so that you can prepare and watch it before your lab section for the people on Tuesday. Or at the very worst, at the very beginning of your lab section. I guess that would be okay too. But I'd like to go quickly through what our project is like because I'm still setting it up, and it's a little bit in draft form. So if you run into problems, that's why I want you to be doing it in the lab, because you can talk to the TA can talk to me, I'll be in my office hanging out waiting for things to go wrong. But with that said, I'm pretty excited about what we can do with this project, and so I want to tell you real quick about what it's going to be about. It's a bioinformatics project that we're doing entirely on the computer, and we're going to predict two things: stability and antigenicity of microbial protein fragments. By stability, we just mean how well the protein folds, it doesn't precipitate, it's well formed in solution, it behaves itself so that we can do biochemical tests on it. By antigenicity, all I mean is antibodies can bind it. Antibodies are the proteins that your body makes specifically to bind microbial pathogens. And so we want to know how those antibodies bind a microbe, because we have a slippery microbe, a microbe that is evading the immune system by changing its coat. Because of that, we want to understand how it works so that we can get around it and we can possibly treat the disease better. First things first, all the detailed protocols are not in a PDF protocol like usual for the other 4 labs. They are on genieact.org, and there's going to be links to that. I'm going to get them all active tomorrow morning, and we're going to have the most updated protocol will be there. If we have a problem in the lab and I need to fix something, I'm going to fix it on the protocol. Because you're doing it on the computer, it'll be real easy for you to refresh the protocol and bring up the corrected protocol. So the thing is, the online protocols are there. If you're having an issue, do try refreshing, because it's possible that I'll have changed it in the time that we've talked. And we're going to post a pin on Canvas. At the end of each part, there are 5 major parts to this procedure. At the end of each part, make sure that you go through and submit the text and the files to Genie that we have. We're also going to have a data entry sheet that will contain all of your data in one place for you and for me, and so you'll be submitting that through Genie as well. But just realize that you have 2 ways to input data. Genie itself, you enter the field on it, and also put it into the data entry sheet that we have, an Excel sheet. So at the beginning of the lab, open your assignment sheet. I will have your assigned clinical sequence for you. I'm going to describe what that is. And then also I want you to sign into Genie and make sure that you can see the detailed modules. In the past, this has required a little bit of back and forth where you sign up and then I click something and then you sign up again and click a few more things. I will describe that completely when we get to it. It's going to be a bit of a hassle, but that's why it's important for you to be active at the beginning of your lab section so that we can do this back and forth. Once you can see the detailed modules on Genie, we are set to go, and you are free to run forward and to answer your question. So the proteins that we're looking at are part of an infectious pathogen that is Mycoplasma genitalium, one of the smallest bacteria with a small genome, and yet it's very crafty despite its small size. It's actually a sexually transmitted infection, so it is very hard to get rid of. And so we want to figure out how to get rid of it. It develops resistance pretty easily to a lot of microbes, and it also evades our immune system. So it's small but sneaky. Okay, so there's only one approved method for its detection even. It's hard to detect and work with in the lab. It's difficult to study because it's slow growing. One of the things our collaborator has done, and our collaborator is Gwen Wood's lab over at the UW. She also works with Patricia Totten over there. But she's Gwen Wood, and yes, she is married to Dr. Derek Wood. I'm going to try to keep the 2 distinct by calling it the G. Wood Lab as opposed to the D. Wood Lab. But G. Wood Lab, Gwen Wood is one of the world's experts on growing Mycoplasma genitalium and on seeing what its clinical effect is. She's collected some clinical data from its infection for humans and how it changes over time that we are basing our project on. We are the biochemists here, so we come in here and we have ideas about how to manage the proteins, how to make good proteins, how to measure their binding. She gives us all of our importance to our proteins, the relevance to them. So how do we understand this? This is a pathogen that infects and persists in humans for a long time, and it does so by switching out some of its genes. It does recombination events to change the proteins on the outside of it, so the antibodies get used to one protein on the outside, then they switch it out and the antibodies can't bind anymore. That's the idea. There's a major complex on the outside of Mycoplasma genitalium that it cannot do without, because it has to do with how it attaches to the body, and it also is where antibodies attach to it. It's a large protein complex made of two chains called MGPB and MGPC. Now, MGPB is shown here. It's the larger of the two, and it has these genetic regions that they could find when they could just look at the genes and see what varied when you have this infection persisting over time. And they saw that three regions, shown here as B, E, F, and G, three regions would consistently be varied over time. It seems like these were regions that it could recombine, and it could switch these out and do mutations in them to avoid antibody binding. So we want to understand, how do these regions bind antibodies or not bind antibodies when they mutate to get rid of the antibody binding? So those three regions, colored red, green, and blue, are going to be on future slides. And then there's a yellow region called the C4A region. It's all the way at the C terminus. It does not change very much, and it seems to be important possibly for receptor binding, possibly for immune function, and antibodies to it are not evaded. So we're interested in that one as a part that does not change as easily. Maybe we can try to figure out, why is this one not able to change when all these other ones are able to change? Maybe this is its Achilles heel, where antibodies can bind to it and then recognize it, and the immune system can kick this nasty bug out of our body. So the thing is, the figure that I showed you was made back in 2015, back before we knew all we knew was the gene structure. Then we have the crystal structure came out in June 2020. This was a big advance for us, because now we could see where all of those amino acid residues were. So this is the MGPB protein that we just showed you on the last one, and so we finally had a structure of it. Notice how we even see how it binds to the plasma membrane of the bacterium, so it sits on the outside, and it has these parts, especially the parts up here, are sort of exposed, right? And so they have all the structure. There's a beautiful secondary structure. This is the entire MGPB chain. It's very exciting to be able to map those genetic sequences. And so the question is, we have these three variable regions. Where do they map out on this surface? And the one C4A non-variable region, where does that map out on this surface? So one thing to keep in mind, this works as a dimer, and so you have the MGPB shown in blue on the left here. MGPC is shown in yellow. And notice how MGPC is kind of the same size, but it's not as big. It doesn't extend all the way down to the membrane. But of course, it's an important part of the complex, and as they show right here, it might be involved. They both cooperate with binding the target cell that they bind, and they probably they probably work in actually a heterotetramer, but we're going to focus on the heterodimer right now, because that is that's complicated enough. So here's MGPB. When we took the structure, we could map out the variable regions, region B, E, F, and G, in red, green, and blue. And you can see that those, they form sort of regions, but they're sort of stretched out and spread out throughout the molecule. When we take those out of the context of the rest of the molecule, we can make them and do some antibody binding tests on them. But they might not be happy without the rest of the protein around. Look at the yellow C4A region on the very bottom. That one is the one that doesn't change, and excuse me, we've got antibody regions to that. So MGPC, in yellow on the last slide, would bind sort of on this side of MGPB, and it's rotated 180 degrees kind of this way. It would bind on that surface. Notice that the MGPC surfaces, they don't bury much of the variable region. It looks like MG is varying the exterior targets that antibodies will bind. It doesn't vary the BC interface, MGPB, MGPC interface right here very much. But even so, if you look right there, you see that there's a little bit of region B that's covered up. I wonder if we really need that. Do we see antigenic variation there? Maybe we can cut off that end of the fragment and make a fragment that behaves better in vitro in a tube in our lab. So that's exactly what Gwen and her lab did. Dr. Wood and her lab went, and you see that this was received pretty much as soon as the crystal structure came out. They started mapping their results onto the crystal structure. Here we have the same 3 variable regions from MGPB in the same 3 colors, red, green, and blue. And then we have the MGPC gene, which follows MGPB on the MG genome right there. That's what typically you do if you have 2 proteins that work together. The KLM region is shown in purple. So depending on which region, I'm going to give one day region B sequences. One day in the lab is going to get region EF sequences, and one is going to get region KLM sequences. So when they looked at 4 different patients, and they sequenced all the genomes of MG that was infecting these patients at 2 different visits. And these patients were treated with antibiotics, and yet it didn't seem to get rid of the infection. So these infections are both resisting the antibiotics and they're evading the immune system. So they have visit 1 is shown in gray. They get a bunch of different genomes because, of course, you have a lot of different bugs that are mutating at different rates and trying different things to evade the immune system. But you notice that you have here the 4 hypervariable regions. And what you have is you have some mutations that show up. If you look on the bottom, the yellow is visit 3, which is 1 to 2 months later after visit 1, which is shown in gray. And you see that there are a lot more mutations. The colors are where that region has mutated. And there's a lot more mutations in the visit 3 yellow sequences at the bottom. These are probably ways that they're trying to avoid antibody binding. So we want to try to get those in fragments that we can make in the lab. Notice also here that region G, the blue one, it's got some mutations, but they're not very much. So it's not very interesting relative to the other ones. And that's why if we have 3 lab sections, well, let's go after the 3 interesting regions, B, E, F, and KLM. If you zoom down on which amino acids are mutating, you can see that you've got a handful of amino acids that are mutating in B and in EF. In G, you only have 3 amino acids that are mutating. So let's not bother with G. Let's look at some of these mutated sequences. Where are the mutations, and how do they change the chemistry of the surface that the antibody will be binding to and recognizing? Like if there's a big charge and they mutate it away, then maybe you can have a hypothesis that that is how they avoid antibody binding. They take away a charge that the antibody binds to. Here's the purple sequences from KLM. You see a lot of mutations here, but which ones are important? Where do they fall in the structure? That's what you can map out by taking these and mapping them onto the structure. So they already did a little bit of this, and if you look at, this is MGPC, and this is MGPC for visit 1 on the left, visit 3 on the right. It shows in black are where there are amino acid mutations that this particular patient had mutations in. And you see that the black residues are shown, they're sort of on the top, they're on these exposed loops, they're exactly where antigen variation would be expected to cause antibody binding to mess up. So that's what we have right here. We have this, this situation where the chemistry of the surface might be changed by these. So we want to go look at the surface, look before and after. Before will be the crystal structure of MGPB or C. After will be whatever the fragment of the gene is that you have mapped out onto that crystal structure. And just to show you that this is probably going to be a very fruitful mode, here's some fresh data from Suzanne Stafford that shows a tale of two domains. Over time, we're taking antibodies that developed over time. They're from two different species. There's some complications here, but the main story is to compare yellow domain 4A to red domain B. It was red in the other ones. We made it blue in this one. Sorry about that, but you can imagine that it's red or blue. That's domain B over time. As the primate makes more antibodies over the weeks, you see that week 2, the antibodies bind more towards the southeast. That is better binding antibodies. These are actually on rates and off rates that divide up to give you a dissociation constant, just like we talked about in class. So Suzanne found that week 4 and week 8 are more toward the southeast on this graph, which means you have better antibody binding. Antibodies develop to bind better to domain 4A over time. But what about domain B? If we take rabbit and we take the before infection and then after week 8 of infection, the antibodies at week 0 actually bind better than the antibodies at week 8. The mutations that are going into domain B over time are causing it to bind antibodies less well. That's what we predict the mutations that we give you are going to be doing. And so this is what we hope to do, is to make more fragments and test them for binding against antibodies and see if we see this pattern for the variable regions, that the binding gets worse. So the Wood Lab has made a lot of different protein fragments, and they've had success. They put different protein tags on them. You have to put a tag on it to be able to purify it with a simple affinity column. And so we either have histidine tags or we have sumo tags. You'll be able to look at that when you get into lab. And we found that the sumo tags help it to stay soluble. And they found that from the fragments that they made, the B fragment works really well in the tube, and the 4A fragment works really well. I just showed you the experiments from those. The reason why we haven't looked at more fragments is partially because when they made the other fragments based on the gene sequence, the variation that they saw in the gene sequence, they didn't get good fragments out the other side. They would crash out of solution. They wouldn't fold right. And so HIS-EF and KLM, they tried to make 3 different KLM fragments, and none of those were very soluble. So let's see if we can improve that. So what you're going to do, these are the steps that you're going to do. Each of these parts goes with a different part on Genie. There's 5 parts that are going to show up on Genie, plus a pre-survey and a post-survey that ends up being 7 parts. But the first experimental part is you're going to take your gene sequence, you're going to look at it, and you're going to compare your gene sequence that's going to be from region B from one day, region EF from another day, region KLM for another day. You're going to compare that to the crystal structure sequence and to the expressed fragment sequences that the Wood Lab has tried from this last slide. So what you're going to need to do is you're going to take the primary sequences, you're going to need to do a sequence alignment, a multiple sequence alignment, and we've got a great program for doing that called CView. I want you to use that this week in lab, and then I want you to compare your clinical sequence to the structure, the sequence from the structure of the MGPB, MGPC complex. So for example, you can do different blast searches. I'm going to ask you at one point to do a blast search against just proteins in the protein data bank, because I want to know if there's other crystal structures that might show up other than our Mycoplasma genitalium structure. In fact, I know you'll find a structure, I expect you'll find a structure of Mycoplasma pneumoniae, okay? But you probably won't find much else, but you need to look. So you're going to blast your primary sequence, but you're going to do it only against sequences that are in the protein data bank that have solved structures. All of them have accession numbers that are PDB IDs, which means they are this 4 alphanumeric character code. Then you're going to get all your sequences and you're going to put them together and you're going to align them. And so the protocol tells you exactly which sequences to do, and you're going to use this program called CView. This I want you to do today. CView is a really simple program. You can actually run it from the download folder, at least you could last year. So we're hopefully going to do that this year. You can literally just copy and paste the text of your sequences in FASTA format in, and then you press the button that says align. And it will do this. It will put gaps in to make the residues align. Each chemical type of residue is a different color. And so if you see the colors aligning, you know that the residues are aligning, at least in similarity mode, maybe not identity. The important thing about this is it's really important for you to probably use the cursors rather than the mouse. Because, for example, here's one sequence. This is a cysteine in position 65. Now notice that when I have my cursor on that cysteine, it tells me right here at the top that it's position 65 in that sequence. It's important to go from the second number. The first number is the absolute position, which means the position with all the gaps in there. The second number is the true position in that exact sequence, and that's what really matters here. This is the 65th residue in this particular sequence. That's the number that you want. So you want to go from the second number, not the first, and that's what's going to make it line up with the PDB structure that you look at and everything. So just remember that. And if you want to know which residues line up with it, then you just press the up arrow from here. And you go up to the next residue up. You see it's also a cysteine, but it will tell you what number it is in that second part right there. It'll say it's cysteine 93 or whatever. In that sequence, that's the residue that lines up, that is similar to the other sequence. So this is really important, and it's really an important tool. Try to use this in the lab so you can ask questions, get help setting it up. Make sure that this works for you. The other thing that you want to do is that you're going to, basically from that alignment, you're going to design a sequence. You want to design a sequence that, has a good chance of being soluble, is kind of a compact fragment, or at least not completely extended like a piece of spaghetti. You want it to include the mutations that are in your clinical sequence so that we can look at them. And you want to try to develop a different fragment, a different start and stop to it. This is what I want you to do in lab, because I want you to get help with this. Once you do that, I want you to analyze the primary sequence, then build a model of the tertiary structure of it. So part 2, analyzing the primary sequence, this is where you take your designed fragment, you copy and paste it into the different tools. And you're going to analyze physical properties and even secondary structure. Now, of course, these are predictions. They aren't necessarily very good predictions, but they are going to be predictions from the primary sequence of what the computer thinks based on that. You're going to do better than that as you go along, but it's a good place to start, and it's a very easy way to compare a lot of fragments. So as you go through this part, as you collect data, I want you to fill out the Excel file that's titled Data Entry Sheet. So you want to go to there, and the way it works, everything you should be filling out is in one column. It has yellow squares. Overwrite the yellow squares with your data. Then I'll be able at the end to copy and paste everyone's column into one big sheet, and I'll be able to look back and forth. So you have like the different information, and the one thing is Excel cells can hold a lot of information as long as they don't have a carriage return, an enter sign in them. So you can make really long, you can paste your entire protein sequence as long as it's just one line without any paragraph returns, carriage returns, enter return, whatever you call it. So notice that down there in step 1, I have you put your FASTA sequence, you have a header at the top of it, and then your entire sequence can be in one line, and you paste it into there. It's going to overlap that box, but it will all be there, and I'll be able to retrieve it from that. So then you'll go through and you'll make physical predictions. You're going to predict what the PI is, what the molecular weight is, and most importantly, what the solubility is. You're going to predict what secondary structure is, and you're going to paste those all in. I have examples to the right over here of where we did this for the Gwenwood Lab fragments that they already made. Again, remember that the first 2 worked well, and the last 4 did not work well. And you notice that this one right here, KLM1, was predicted to be insoluble. So this tool doesn't like it either. But the tool likes the other ones just fine. It says they're soluble. You've got to add up a bunch of tools to try to figure out, are these really going to be soluble or not? So that's what you can do based on just primary structure. Now, you should get to by the point that's really important, that by the end of week 2 in lab, that you take your primary sequence that you've designed with your mutations in it, they're only yours, and you paste it into the Itasca homology modeling program. This program can take 2 to 3 days, 48 to 72 hours. So it's really important that you don't put this off until 2 days before the lab report is due, because then there's no hope of you getting it in on time, right? So start this today if you can. Get to the protein design part. Talk with us about it, and even if you don't finish the previous part with all the primary sequence analysis, make sure you go ahead and paste it in and go forward with building the homology model. What it will do is it will actually take all those residues and it will model them out down to the atoms, and it will allow them to move around. It will calculate what is a better rotamer conformation. You know, it will twist them around a little bit. The backbone is going to be pretty much the same, but the side chain rotamers may be different. And so it's going to be a much more accurate surface than just looking at the secondary sequence or the primary sequence by itself. This is what Itasca looks like. You should be watching this today. And so if you look at this, you go to Itasca standalone, or actually it's down here, it's on the web, and you paste your fastest sequence of your designed protein fragment into there. And actually, that just reminds me, there's some things that I need to make sure that you do. I'm going to write a little note to modify my protocol tonight as I'm working on it. Submit it today or tomorrow. It's not hard and it doesn't take long. Expect it to take at least 48 hours. Some years it actually gets hung up and we have to go to another program. We'll deal with that when it happens. For right now, try to get ahead of the game, and when you get it back, you are in business, because everything else is smooth sailing from there. So Itasca thinks it's going to give you some measures of model quality. I want you to get a second opinion. There's a program, you can plug your model into it, and it will say, oh, I'm going to give your model a score right here, and I'm going to give your model even a residue by residue score. I think your model's better at these residues, and it's worse at the ends. That's sort of normal. But if you have a weird part in the middle of your protein, maybe you want to think about taking that model with a grain of salt. So, like, here's where you get the score from this Voro MQA model. And then you are ready to take the PDB file. At this point, you have a PDB file with the XYZ coordinates of all the atoms in your fragment that's predicted by Itasca to be the structure of your protein. So now you're going to look at that structure, and you're going to be going off the structure rather than just the sequence. This is really powerful. So the first thing you're going to do is you're going to take your structure in PyMol, and you're going to align that structure with the structures of the other things that we were looking at, including our crystal structure, MGPBC, which is 6RUT is the name of that. So for example, what I've done is I've done this real fast with an example of the, actually this is the week 8B fragment that Suzanne measured the binding of in the other thing. That's another story. But you see here I have MGPB in blue, MGPC in yellow, sort of with the color scheme that the other lab was doing. And I have the little C terminus of it. You see how it sticks out like a little comma. This is the part that binds the transmembrane region. This is how I want you to look at it, with this part down and then rotate it around so that you can see the different parts of the surface. Paint it different colors, make different surfaces, try to see where the mutations are and how they will change the chemistry of the surface. So orient them like this, and remember that there's that very useful sequence mode. You can go through here and you can click on all of your mutated sequences, all your mutated residues, and you can make the selection out of them and color them all one color. You can make them space filling so they stand out. You can do all sorts of things. Remember all the stuff you did at the tutorial week? That was for a purpose, so that you can do this on your own, training wheels off this week. Training wheels not entirely off because we're providing help, but that's why you need to be active during your lab, because that's when we can provide help right away. So you're going to use PyMol to do some calculations after you do the alignments. You're going to calculate the accessible surface area of your fragment, and you're going to use two programs, VAST to do a structure search, and AgriScan 3D to calculate aggregation propensity from the structure. This is actually really useful. This is what the output of AgriScan 3D looks like for the B region fragment that I've been working with. And you look at this, and you see after 8 weeks of mutations, this is the B region fragment that we were looking at. And remember that the B region fragment works well for us. It's very stable. Even though it's really elongated, it doesn't look like it'll be super stable. but I think that this core is big enough that it behaves well in solution. But notice that even this, the best one that we have, it has this red patch on the bottom. It has these blue patches on the outside, which probably are some of the reasons why it stays soluble for us in the tube. This red patch right here looks nasty, and if you look at it, it's like pointing into the main chain of the protein when you line it up with the crystal structure. I wonder if we could choose a fragment that didn't have that red or minimized it somehow. So that's what you can do in your fragment design. You can choose where the fragment starts and stops, and you can run these tests to see if your design fragment is predicted to be stable in solution. Does it look like that picture I just showed you, or is there a lot more red? If there's a lot more red, we might want to go another direction. And by the way, yeah, you don't have to design a good fragment. I just want you to tell me that your design is predicted to be soluble or insoluble. And everyone can also do the second question. Remember, our second question is antigenicity. How do those mutations that were collected from patients in the clinic, how do they change those exposed surfaces that the antibodies bind to? One of the things you can do in PyMol is that you can measure your fragment. So there's you can select atoms. This is where you're going to use the pick atom function. Look down in the lower right-hand corner menu, and the PKAT, picking an atom rather than selecting it, is what you'll want to do. If you do this, you can measure the distances between two atoms, and it gives you the distances in angstroms. So you can use this as a ruler to measure how long is my fragment, how wide is my fragment, how close are my N and C termini. The end result is at the bottom of the data entry sheet. I let you interpret. Answer the questions, sum up the things that you've seen. And so, the important dates. This week is week 2. Make sure you're in lab. Week 3, lab will be open as well, but also, you can also try to do some of that remotely. Lab is only open during the times when we have people in lab. Usually there's room, because usually everyone can't show up for every lab. And we'll work it out if we need to find room for you. So just remember, if there's one thing I can say, please don't come to me in a week and a half and say, how do you submit Itasca again? Please come to me this week with how do you submit Itasca, because it takes 2, even 5 days before it comes back. You need to allow yourself time for week 3 to do all your cool structure stuff. Reports are due in 3 weeks. Friday, November 20th, a little less than 3 weeks. And that's when data is going to be due to Genie. The data entry sheet will be submitted on Genie. I'll be able to see it, and I want to see your interpretation. Then we'll go through all those, and we'll sort of decide which proteins will we make next quarter in lab. And those of you taking lab next quarter, if I can, I'd like to give you your fragment if you find one that you think will work well. So there's a definite prospect for that. All right, but that's a whirlwind tour of what you're going to be doing in the lab. I'm going to let this process, and I will hopefully see you in lab or in my office this next week.